Good evening. It is the 19th of March, 2026, and we are doing Journal Club for the Science Academy at Holy Cross. And we are covering a paper that came out in November in the journal Nature. And it is rare genetic variants confer a higher risk of ADHD and implicate neuronal biology. And this is Demontis or Demonti et al. And this is Ciara and Connor are the facilitators.
Hi, my name is Ciara Proulx, and Connor Hodgkins is my other facilitator today, and our paper is on the rare genetic variations that have a high risk of ADHD in neuronal, how do you say that?
Neuronal?
Yeah,
Yeah.
biology. Okay, first off, first we want to do an introduction to ADHD. I think everyone pretty much has a basis of what ADHD is, but we just kind of wanted to go over it just in case. So, ADHD, the long-term form is attention deficit hyperactivity disorder, and it's a developmental disorder that's usually known by people having inattention, hyperactivity, and impulsivity. So, these are things that are such as like moving around a lot, feeling like restless, talking excessively, interrupting, intruding, difficulty paying attention, a bunch of stuff like that. And um, there's a lot of like ADHD is very common, and so is ADD. So this paper, the focus of it is there are a lot of papers on ADHD genetics, but they're all on the common genes. So this study looks at like the rare mutations and how they contribute to ADHD and the related disorders such as like dyslexia, dysgraphia, and dyscalculia. So,
So, how does ADHD actually work in the brain? So, ADHD is very closely linked to lower levels of dopamine, and because dopamine levels are lower or less efficient, it becomes harder for the brain to stay focused, especially on tasks that are not immediately interesting. Also, ADHD involves inefficient neural networking, and that means that the parts of the brain that are responsible for filtering distractions and managing time don't communicate as effectively as they should. And as a result of that, people with ADHD often experience like chronic difficulties with attention regulation, meaning that they may struggle to stay focused, complete tasks, or manage their time. So, yeah.
okay, so there's a couple parts of the brain that ADHD affects. So it affects your prefrontal cortex. So as it says on the screen, this is the part that has all the executive functions, planning, organizing, paying attention, your neural networks. So the it creates differences in like how certain parts of your brain are going to communicate with others, which is also something that happens in dyslexia. That's the part of the brain that people who are dyslexic have trouble with. And the striatum,
Striatum.
striatum. So that's like your reward, like part of your brain and the part that gives you motivation. So, people with ADHD tend to lack, like care for, getting rewards or being reward motivated. Like a couple studies have shown that, and they talked about that a little bit in our paper. But we just wanted to go over the parts of the brain and exactly what ADHD does for it. So this is figure one. Does anyone want to, take a stab at it?
Don't be shy.
Yeah, don't be shy. Don't raise your hands all at once, guys. I don't know who to pick. Okay. So figure one. So what this is showing is it's the comparison of like the mutation counts in people with ADHD and the control group. And the control group is people without ADHD. So basically what this figure is saying, people with ADHD tend to have more of these rare mutations than people without ADHD? Char.
Correct.
Anything else?
Well, okay, so I'll insert some. Go back, uh, go back a slide.
I don't even know how to go back
Use
because
the
I'm
arrows.
clicking on my screen, my friend.
Can you click
You
the
want?
left arrow key?
No, I know, but it wasn't letting me do that earlier.
Oh, um. So this paper's not really about mutations. We're gonna use the word mutations as kind of shorthand for what it actually is. What happens with mutations is that when you're copying DNA, it's not perfect. Like if I gave you a book, you actually see this in books like the Bible, where the Bible originally was just copied by monks by hand from person to person. Like a monk would copy it, then give his copy to someone else who would then copy that copy, who would then copy that copy. And if they made any mistakes, which of course they would because they're human, it would build up over time. That mistake, when it's made, is called a mutation in DNA. But if it sticks around after several generations, it's not referred to as a mutation anymore because it's not new. Mutations are by definition new. So instead, it's called either a variant or a polymorphism. And so there are, as Ciara said, there are a large number of variants that have been linked to ADHD. I'm in the process right now pulling up my genome that I got sequenced 2 months ago because I noticed I was actually positive for a number of those genes. What's novel about this paper is they actually discovered a whole bunch of new, not mutations, variants that are linked to ADHD. And so if you go to figure one again, then what that's looking at is um, kind of hard to see because if you've been paying any attention in bio, the error bars are gigantic. Um, Ciara,
Hmm.
I didn't think they did a good job of this. So I'm going to ask you to clarify something. Can you clarify all the acronyms?
All the acronyms.
Yeah, RPTVs.
Um,
So like
RPTV.
RPTVs. Like what is an RPTV?
Isn't that
What
like the
is that?
proteins? Aren't those the proteins in like the sequences?
I'm gonna see. Hold on. I gotta do a search. RPTV. Uh, what happens when you submit a paper and it takes a year and a half to go through the process of review? Sometimes the introductory stuff you originally had in the paper gets taken out, or you add new stuff not realizing you didn't introduce it, and so this becomes a little more difficult because you've got all these extra names. They are rare protein truncating variants. Okay, so they're the variants that we're talking about.
So I was like
Okay.
sort of right.
Yeah. So then what's arsin?
An arson.
Yeah.
Um, is that like the sequence the proteins are in
Rare,
that are like
it's
damaged?
rare synonymous variants. It's a control. That's what it is. Okay. All right. So, freshman, you're not going to get this. You're not going to understand, but I'm going to mention this probably in about a month. I will mention this thing and be like, remember when we did that paper? Okay. But for sophomores, remember last year how we talked about where proteins come from? Right? You have a gene, it's a recipe. And so the gene is a code and it's written in words that are 3 letters long. Do you remember codons? Yeah. Okay. Those codons are on that codon table that's in the back of my room. And you go and you say, okay, I've got these 3 letters, UUC, right? And you can look up UUC and see what peptide, what amino acid it is. A synonymous change is when you go from like UUC to UAC and it doesn't change the amino acid. But you'd have to be looking at that really complicated chart that I haven't memorized. So what I just said may not be true. I'd have to pull up a codon table to see it. But a synonymous change means no protein change. Even though it changed the DNA, it didn't result in any type of amino acid change, so therefore the protein's fine. That's a control, a negative control. Do you see? And then for then the ones on the left are the the far left are these new rare um, polymorphisms.
Are you
So
talking
then
about A or B? You said on the left,
I'm
right? So
talking about
A.
A. Yeah, A.
That's what I thought.
Arsins, and then so what's a DMV?
A DMV.
Yeah. So
Didn't you?
guys, when you're presenting anything, you always want to orient the reader to any acronyms, what's on the x-axis, what's on the y-axis. You want to tell us all that. So like, what's a DMV? Huh. What's a DMV? No exact matches. I
Department
f
of Motor Vehicles.
okay. So what if I say severe? R severe. Okay, there they are. They are rare, severe, damaging missense variants. Okay, so I told you the control is a synonymous variant. Like I'm a geneticist. I've never seen these acronyms before. So this team decided for ease that they would refer to them as DMVs, um, our sins, our PTVs. In the field, we don't really do that. So this is their own like lingo, which is why I have to look it up. Um, so this severe DMVs are variants that are missense. So if a synonymous change does not change the protein, what do you think a missense change does?
Can you repeat that?
If a synonymous, you know what a synonym is, right? Oh, geez, I
Yeah,
don't know if you know
like
what
a
a synonym
similar.
is. Yeah,
Yeah,
but
it's like a similar word.
right, do the freshmen know that? Freshmen, do you know what a synonym is? Not cinnamon. That's a delicious spice. But a synonym is just another word that means the same thing, right? So a synonymous change in your DNA leads to no change in the protein, same result. But a missense means that it's actually creating some other protein. Okay. And severe means obviously what it means. It's that it ends up being very bad versus a moderate one. And so what this is showing, and I believe this is statistically significant, though they don't have anything here, is that there is a relationship um, between those severe DMVs and autism, which and I think later on they also say not autism, sorry, ADHD, but later on they say also possibly autism, but it was unclear to me. I couldn't really understand it. Uh, Look at how big the study is. Like if you look in B, where it says control and ADHD on the x-axis, look at how many people. Wow, wow, 9,000 people per group that they did a whole genome scan. That good enough?
What? Yeah.
That good? Yeah. Okay.
So figure two, does anyone want to present figure two? Oh, Dylan, perfect.
Go ahead, Dylan.
Okay. Um, figure two. We saw in figure one that people with ADHD have more of the like rare variations. So this figure 2A is really saying like which of these variations would increase your risk for ADHD. So the ones who well actually I'll start. The y-axis is the basically just like the significant how much um more these genes show up um in people with ADHD versus just like a control. And then
I'm going to
next,
interrupt you for a second. I'm just going to interrupt for just for a second because I want to. This can get confusing unless you're using precise language. Every, excuse me. Everybody has these genes.
Oh yeah,
Everybody has the genes.
variation.
You're talking about not variations, variants.
Okay.
Variants.
Mm-hmm.
Variants, or polymorphisms, whatever you want to call them. But that's what it's listing a whole bunch of genes that everybody has, and what it and okay, you can explain the rest, but I just wanted to make
Yeah.
sure we were clear about that. Okay.
Okay. So yeah, it's just the different variants and which ones are more prominent in people with ADHD. So the ones that are higher on the y-axis means, of course, that they're more like more in ADHD people with ADHD. So first you could see ANK2. So the three main ones that are over that like red line, ANK2, MAP1A, and ANO08. So these are like the three variants that they found are the most like significantly like more in people with ADHD. And it actually makes a lot of sense if you look into what these genes, what like variants in these genes would do. For example, ANK2, it's makes like the protein uh ankyrin B. And if you didn't know, people with ADHD have a much higher risk of developing cardiovascular disease. So, and a this like a variant in this, the mutation in this um protein could cause um higher chance of cardiovascular disease. So that's kind of like linking, and it makes a lot of sense. It links why ADHD people with ADHD tend to develop cardiovascular disease. Then the next one, MAP1A, a mutation in that, so it encodes a protein that helps stabilize neuronal microtubules. And a mutation that could impact development of dopamine pathways and serotonin. And like Connor said before, that's what the main like thing in ADHD is. Like difference like in dopamine receptors. And then lastly, ANO8. So alterations in this cause something called ICP, which is a liver disorder in pregnant mothers. And it's actually found that children born of these mothers are have a higher tendency to develop ADHD. So it actually makes a lot of sense why they would have that gene mutation. And then figure B is really just showing uh, so y-axis it's OR. It's basically just saying how much carrying that gene variant increases your risk for ADHD. And then the x-axis is just like how rare or common. So to the left would be like very rare, and right would be very common um, alteration or variant. So as you can see, to the left, um, EIF3 G. So that shows that people. So it's a very rare gene. People that have it have a very high risk of having ADHD. And then you can see those main ones that we were talking about, ANO 8, MAP1A, all those like they have a higher risk of developing ADHD, but they're still like on the rarer side. And like all the red ones are rare. I'm not really sure what CNV means. Yeah, I'm not really sure. But and then yellow would just be like the common ones. And obviously the common ones would not have that much like significant difference in whether or not you have ADHD. So in short, it's basically just saying which genes are really like uh causing like ADHD.
CNVs I'm actually familiar with. That's copy number variants. So, um, how many copies of a motif within a gene are there? So, in Huntington's disease, for example, people who have Huntington's they have this long sequence of the same. I think it's I want to say it's 3 bases, and it just keeps getting more and more. It keeps the polymerase keeps accidentally making it and making it and making it and making it and making it, and so you have multiple copies of that particular motif. So it's a copy number variant, and it actually tells you where that copy number is because it's usually chromosomal. So 22q means the 22nd chromosome, Q arm, 11th band, region 21, and it's a deletion. That's what that means for that top one.
Thank you, Dylan.
Mhm.
Does anyone want to do figure 3?
All right,
Carter.
Carter.
Okay, so bear with me here. I tried my best. But from what I understand, the first panel of figure 3, each of the little dots you see is a different protein group within a gene. So the pie charts on the right, B, C, and D, are basically each showing like so it's showing which proteins are supposed to be in that gene group working together. And then the smaller like slice of the pie chart is showing which ones didn't fit within that group. So, and then E is showing how they tested all three different gene groups. And the little, I don't want to call it a scale, but basically the darker that a square is, the more often the rare variant risk genes show up. So they also tested it against autism, developmental disorders, and schizophrenia to see if they overlapped because they have similar brain patterns. Yeah. I don't know if you want to add anything onto that.
Do you know, sorry to like put you on the spot, but can you maybe explain the like what B and C are? Like, do you exactly know? Like, I
I
know
don't know.
it's the index proteins, but when you were like looking at the figure, did you specifically like notice anything about them?
I mean, I didn't like notice anything specifically. I kind of just got that from it, that it was trying to test each one and showing how often they showed up and which ones were supposed to. But yeah,
That's
and
that's sounds right to me because it was done as a search. I think you're right, Carter.
Yeah, Carter.
thank you.
All right, who wants to do figure number 4?
I could try figure 4.
All right, go ahead, Juliana.
Okay, so panel A, we have the blue line, which are the top 17 genes of ADHD risk, so like what they think causes it. And then the orange line are all like the other genes, which they call like the background genes. And they're just comparing to see like which are more expressed like across like your age, because like at the bottom it measures it so you could just see like where are the ADHD genes like more prominent, like when do they typically appear. um so it's basically what you can take away from this is that the ADHD rare variant genes I think are more active, so they like appear more throughout brain development. And then panel B, I don't know what these like the things stand for on the side, like the
Those are the genes.
MA. Okay, I just don't know if they like each letter what they mean.
Oh
But
no, because those are the gene names,
okay.
the names. Yeah.
So panel
So
B is
those are the
like
ones we've been talking about. Those are the genes we've been talking about.
so
So
panel B has the same Y and X axis, so we're still like measuring how these genes appear across all the different developmental stages. And the MAP1A, I think that's one, appears to be like really high, like after birth, so like later in life it starts to go up. And then the AN08 is higher before birth. like it starts decreasing. And then ANK2 is like probably like the most stable of all of them. Like there's no sudden like increase or decrease aside from like two of them. So it basically just means that each ADHD gene has like a different developmental pattern. And then panel C, all the colors. It's just used to show like the strength of each gene. So the red has a high SCDRS score, which I think it said like it's something, um, I don't remember what they said the abbreviation was. I don't know if you know Dr. Chart, but
I mean, I think I know what this is, but again, they're using acronyms I'm not familiar with.
yeah. So the red is just like um the high, if it's like high, if it's very, very red, the cell is expressing more ADHD risk genes than they thought. And then the blue means that they have low expression or like anything that's like white or blue. And it's basically just to show you which cell types are more affected by the ADHD genes. And that's kind of what I got from it.
Thank you.
Yeah, Dylan.
I have a question about like how they did this. Like how did they find like the genome of like a baby that wasn't born yet?
Um, it's done in,
They they probably took like parts of the cells, right?
well, so the data in C are actually from induced pluripotent stem cells that they have then turned into neurons. Um, so stem cells are just like precursor cells. And so those are human. My impression was that A was on a different species.
Like mice or something. They didn't
Yeah.
really specify what species it was like in the paper. Or maybe I'm just a bad facilitator.
I thought it was supposed to show human brain development, so like why didn't they use a human? If it's
Well,
directly supposed to show like
it's
human stages.
because it's generally frowned upon to execute a baby and take their brain and then take all their cells and
Too
jumble them up
bad.
in a blender. That's why the only time you're going to get a baby is if the baby is like stillborn and or an abortion and donated to science if it's human.
All right. Who wants to do figure five? Any volunteers?
Cold call time if you don't volunteer.
Anyone want to take a shot at it?
You guys take all the fun out of this. It's not you're supposed to be cold. They're supposed to be cold calling for everything, and so that gives you a chance to go. You know what? I just rather get it over with. I'll go, right? Which means you have to read the paper.
Yeah, I can try. I haven't really looked at it yet, but I can try. It looks like it's just comparing people with ADHD and just like their just like education and like social economic status. So I think, oh, what they're just trying to do is like see whether or not ADHD is like caused by like environmental factors. Like if you don't have like education or if you don't like make a lot of money, like if it would like have an impact on it. Um, I'm not sure what ADHD WOLE
It's
is. Is that
it's
like people that like carry the
without intellectual disability. Like
so
why?
they carry like the variant, but they wouldn't
No, it just
like
means
show
that
us.
these people do not have an intellectual. They have ADHD, but they do not have any type of other intellectual disability.
Oh, okay. And then the OR, is that like what would that mean?
OR the whole time has been about ratios.
Ratio of like
It is the odds ratio. What are the odds of that being
of
real?
having a okay. Wait, how would you have ADHD without symptoms? Wouldn't you just not have ADHD?
No, 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 no. Wait, without. Why do you say without symptoms? Would
Wait, isn't that what W
no, no, no.
ADHD?
It's without intellectual disability.
Oh,
So,
oh yeah, okay.
wait, Char, can you give an
So wait,
example of
okay.
an intellectual disability for like everyone?
But an intellectual disability is really just anything that is uh, outside of normal, specifically in the bad direction, like where it's not as good as or with. So freshman, how so you'd be an outlier by what? How do you define outlier? Freshman.
It's like significantly different.
Sure, but what does that look like if you're actually an outlier? You don't even get to the point that you analyze the data. So what's an outlier? How do you define that?
Far from the average.
Yeah, how far? There's a specific.
Significantly.
Yeah, no, stop it. This it's how far specifically to make it significant.
Very far, like. Well, oh wait,
How
oh,
is
wait.
the range?
Yeah.
The
Okay.
two bars you were
Two,
talking about in class.
two. I said in class, I said two standard deviations from the mean.
Oh,
So you find out what the standard deviation is, you multiply by two, and then you subtract that from the mean, and that gives you an outlier. Anything that is less than that's an outlier. Okay, and that's based in statistics and also kind of just an arbitrary number. It's a little bit of both. So this is ADHD without, WO is without ID, which is intellectual disability. That's what that means. So is there a difference between the education, the tendency for lower education in those two measurements? Is there a difference between socioeconomic status? They're trying to make a claim that it's like that they're linked. But as we said in at least what A block, post hoc ergo propter hoc, just because something happens after another thing doesn't mean that it was caused by it. Right? The great
Carter,
legal
you have a
fallacy.
question?
I was just gonna say, so panel B, is that essentially doing the same thing with like each gene and testing it on how greatly it impacts like someone's IQ, for example?
Yes. Um, and what they do you, Dylan, do you remember? Are you still doing this? Do you remember what the drop was?
Um, what do you mean drop?
The IQ drop that they noted.
Like in this figure here?
Yeah.
Um, like
They actually
for
listed
which
it in the
one?
text.
Oh,
Yeah, they said it in the text how far a drop, and you guys might think it's like nothing, but it's like 2 point something IQ points. A drop of two IQ points is a lot. So, um, the United States, for the first time in its history, the average IQ of people your age has dropped significantly for the first time in the history of the country. Now, why might that be the case?
ChatGPT.
AI.
Is it? Is it? You think so?
Well,
Huh.
I don't exactly
Technically,
think AI as a whole. I think the overuse of AI in education instead of just like doing your work or using it not to like better yourself just to cheat, things like
Sure,
that.
sure, Ciara, but doesn't that presuppose that you were given work to do in the first place?
Yeah, but I also feel like some of the stuff people put into ChatGPT is like
I mean,
busy work. Like,
it could be like that middle schools don't really teach that well anymore. Because you keep
Bite your
bringing that
tongue.
up.
Oh, me? I never said anything like that on a video or anything. I never said anything like that. Thanks for outing me, Quinn. Jeez.
You say that every class.
On a video, I'll have to edit that out. But yeah, I mean that's right because Ciara is making the presupposition that there is work to put into ChatGPT. But if you go through middle school and there is no work,
But you
because
also like
boy,
you
we
get work.
really like to make you feel better.
I don't know. I had work in middle school. I have work now. It's not going to change. I'll have work in the future. I mean, I'll have plenty of stuff to put into ChatGPT, but I mean, I still wouldn't do that, but
Okay. Um, these figures are really hard to understand and swallow because they're doing confidence intervals, which is not the type of statistics that I taught you guys as freshmen. Um, and so while there are differences here, there and the p-values are significant, they're less than 0.05. I It's a confidence interval, which is correlation, not causation. It's not a difference.
okay. If that's all we have to say for figure five, Dylan, you don't have anything to add? You did a really good job jumping in there for us. So, for some of the conclusions, we just wanted to say that the study shows that the mutations play a major role in ADHD, especially in the people that tend to have more severe symptoms or other disorders, like the intellectual disorders we discussed a couple minutes ago. And one of the biggest things about this paper is a lot of their research was not on the common genetic variations. They were on the higher impact of mutations. Charlotte, if that doesn't make sense. But they're in the more brain related important genes, and it explains how ADHD can vary in between different people. Does that make sense?
Say it again. The last
Um,
thing you just said.
last thing I just said.
Yeah.
So the mutations and how they're the mutations and how they're different in people kind of explains how ADHD can vary from person to person.
Oh, yeah, sure. Okay.
That made sense?
Yes.
Yeah. So then, one of our takeaways is the genetic mutations can make some of the genes increase the risk of ADHD up to 15%. Um, Connor, do you want to add anything before we get into our favorite words and some of the other stuff?
Now you said it all.
Perfect. So my favorite word from the paper is neuroblasts. I just thought it was fun.
Mine
Uh,
is polymorphism. I just think that's a fun word.
so we thought that this paper belongs in Nature. I think yes, but I think if I don't know if I'm allowed to say this, but if I could change one thing about this paper, I'd probably change the use of oh my gosh, I'm blanking on the word. When you shorten something.
Acronyms.
Yeah,
Yeah.
acronyms, because the acronyms were very difficult in the figures, and then also the way they presented the figures in the paper, like the literal figures, the images were kind of hard to explain. So that would just be something like I could fix. But I still believe this belongs in Nature. Connor, what do you think?
Yeah, it does belong in nature, I think. And journal impact factor is 48.5.
Pretty high.
Pretty high. It's the highest in the world. Um, I'm
I
I'm
don't know.
s-
That's what Google told me.
I'm still stuck on Juliana's question because I
Juliana,
think
wait, can you repeat her question?
Juliana, you want to repeat it?
I don't really know what I said. Was it a
Well, you were talking about figure four,
Yeah.
right? And and you were like, why aren't they testing that on babies? And I think
Oh.
Okay.
I think I'm wrong. I think I end up being wrong here. I just looked it up, and don't I don't understand. So this figure 4C, I thought I knew what that was, and now a top uniform manifold approximation and projection of sRNA-seq data showing clustering of cell types from human induced pluripotent stem cells develop towards midbrain neuronal cell types. Results are shown for cells developed for 11, 30, and 52 days and classified as astrocyte-like, dopaminergic, ependymal cells, ependymal 2, floor plate progenitors, and neuroblasts, which is Ciara's favorite word. Proliferating serotonergic-like neurons with the serotonin transporter, unknown neurons. So this is what all those things are. Okay, so I went to the website brainspan.org. It is an atlas of the developing human brain with a, I should know this is my field, but with a developmental transcriptome. Wait, where are they? Reference atlas. Hold on. I don't know. I don't understand it, but I believe that no humans or animals were harmed in the making of that figure. I think they went and created some neuronal cells in a dish and then tracked the expression of the genes. I think that's what was happening. And so you'll see on the left in C, the left part of C, those are very, very simple, so there aren't a lot of colors. Then when you move to 30 days, the types that have developed from that basic cell have increased a lot. And then 52 days, they're much more constant, but you're not looking, this is not a map of the brain or anything. This is a two-dimensional cluster analysis. I think it's cluster. It's either principal component analysis or cluster analysis to see how they're related to each other. But I can't get any more detailed than that because I actually do not understand it.
So if that's it, unless someone wants to answer that question for Char. No? Oh, oh,
I do that.
actually. Oh, okay.
Why, you had something else?
No, I was just trying to make sure my Zoom wasn't like crashing.
Oh, no, no. No, it was me.
Connor and I just wanted to give a big thanks to everyone who stepped in and helped us present the figures. That was really helpful and that was really nice.
Yeah, thank you all.
And I hope in the next couple journal clubs, maybe this showed you guys that it's actually easy to kind of read a figure on the spot. Well, easy.
And that sometimes you don't. I couldn't. But I do think you thought the paper was good.
Now that we're kind of looking back on it, it's not my favorite. Like I really liked the topic of it, but I feel like it was very confusing. Maybe it would have been like nice if I'd like taken neuro before this.
Yeah, I don't know if you needed that. I think, like, why is it in nature?
Why is it in nature?
Yeah.
Because it's like the nature of our bodies and our brain and
No, no,
exactly
no.
how like
What? How? No, I'm saying like Nature's considered the best journal in the world.
oh,
How
okay.
did how did this paper get in it? What was it about this paper that makes it so profoundly important that it got into Nature?
Maybe it's about the babies in figure 4.
Oh, the babies that Juliana killed. For science.
IQ change?
Yeah, I mean, I think that's somewhat important.
Oh, do you think it's because they studied the things that are like the kinds of genes that are rarely studied?
Yes,
I think
I
that's
do.
probably why, actually.
Yeah. So like to give you guys some perspective, about like how far the field's come. My very first paper that I ever published, I had discovered a new polymorphism that had not been seen before. And we realized that it was going to be a functional change in a major circadian gene. And my genetics advisor, the guy I worked for in my first postdoc, He's like, if you had made that discovery 2 years before, so this was 2006. If I had made the discovery in 2004, the paper would have been published in Science. But instead, it ended up being published in a really good journal for circadian clocks, but not like a widely read journal called the Journal of Biological Rhythms. And that was the difference, was that so many people had already published papers where they're saying, okay, I've got one change in a gene and it causes this little phenotype. But when it comes to you've just found a bunch of new genes, a bunch of genes that have new polymorphisms that haven't been seen before, for something as profoundly important as ADHD, then it gets into nature. Make
Does
sense?
anyone have any questions on that or anything in the paper, like any last things?
All right.
Of course.
Well, thank you guys. We'll go into overtime now. So see you later. Bye-bye.
See you later.
Yay. Good job.
Thank
Yay.
Yay.
you guys.
Thank you.
Editing this is gonna